Let's say that inside this cell, the boss, or the nucleus, is generating a message out of RNA, sending that RNA message out of a little nuclear pore and to a minion, a ribosome right here, which is reading that message and then using that message to construct a protein. This long, winding, contiguous green structure right here is the place where those ribosomes go to deliver the proteins that are in the process of synthesizing, and it is called the ER. ER stands for endoplasmic reticulum, which works hard to recognize partially made proteins, such as the one that this ribosome just put in the ER. When it detects those proteins, it packages them and sends them to the next place for them to be modified and finalized. And it does so in this small budding structure that comes off of the ER and then goes floating through the cell, and that's called a vesicle. So the vesicle buds off of the ER, floats through the cytoplasm until it gets to the next structure, called the Golgi apparatus, for further processing. The Golgi apparatus looks sort of like a stack of pancakes, so you can see as the incoming vesicle puts its protein inside the first pancake, that pancake will mature into the second pancake and so on, and so the pancakes sort of move in this direction, and as they do, the contents change. For example, you can see the protein's shape has changed a little bit, and also now it has a little tag that specifies what its destination is. For example, some proteins could get labeled to end up inside a vacuole, such as this one. Others could be ending up in the lysosome, such as this one. And others still could be secreted out of the cell, and so they would be tagged headed for the extracellular matrix. In any of these cases, the proteins are sent out in their own vesicle again, and then that vesicle will fuse with the target organelle, such as here you can see this vessel fusing with the plasma membrane until it becomes contiguous with it and lets the protein out to where it needs to be. This whole process could be happening, for example, in the breast cell of a pregnant woman. Breast cells need to make milk for the baby, and so the DNA that codes for milk proteins would get turned into an RNA message, sent out. The ribosomes would make those milk proteins. The ER would say, aha, a milk protein. Great, I'm going to send that over to the Golgi by a vesicle. The Golgi says, oh yes, this is a milk protein. That needs to go out of the cell. So I'll put a little extracellular matrix tag on that, and then the Golgi says, all right, great, send that in a vesicle to the cell, and then out it goes so that it can continue out of the breast, and the baby can actually have it. A little side note about the ER, you could technically divide the ER into two sections. There's this one section where the ribosomes are coming by to make proteins, and then on the other section there aren't any ribosomes. Instead, there are enzymes inside the ER that are busy making lipids. These two halves of the ER have different names. The rough endoplasmic reticulum, which is called rough because it looks rough. It has a bunch of ribosomes stuck to the outside of it, making it look all like bumpy and stuff, while the other half is called the smooth ER because it's not so bumpy. So, a quick overview in construction, packaging, and shipping. The rough ER makes proteins, the smooth ER makes lipids. The ER ships partially made proteins to the Golgi apparatus, right here, out of the ER, to the Golgi apparatus in a vesicle. The Golgi apparatus then modifies, sorts, and ships those proteins to their final destination, either lysosomes, vacuoles, or the plasma membrane. Lysosomes and food vacuoles, you say? What are those? Well, they're basically specialized vesicles, so just like the ones getting sent from the ER to the Golgi, they're just a little ball of phospholipids. The thing is, they have some really cool stuff inside. For example, inside a food vacuole, there's, you guessed it, food. And inside lysosomes, there are digestive enzymes, those enzymes that break stuff down. So let's say the cell had a little bit of food that it wanted to break down. Well, it might take its food vacuole and join it with a lysosome, and then the lysosome dumps its digestive enzymes in on the food, and the digestive enzymes break it down until now you have a whole bunch of monomers and things that the cell can actually use, rather than a big old chunk of food which wasn't very useful in the first place. So lysosomes and food vacuoles are specialized vesicles. The lysosomes fuse with the food vacuoles, or sometimes old and damaged cell parts that need to be broken down, and breaks them down using digestive enzymes. In plants and other microorganisms called protists, vacuoles have some other functions. For example, the central vacuole is found in the middle of plant cells, you can see it right here, used to store water, and when it's nice and full, it inflates the inside of the cell wall and keeps the plant cells nice and strong, and so the plant can stay up. If that poor central vacuole doesn't have enough water in it, though, the plant's not doing so happy, and that's because the central vacuoles aren't very full. So central vacuoles store water, and for organisms like this paramecium right here, which live in water, they actually have too much water in their cells. There's water that will go flowing in, and if it keeps flowing in, the whole cell might pop. And so they use these contractile vacuoles right here to squeeze all the excess water out before the cell explodes. Now, how does a cell squeeze? Indeed, how does a cell move at all? Well, that's the responsibility of yet another organelle, the cytoskeleton. You can see this is a picture of a cell where a scientist took some fluorescent dyes and attached them to these long strands that run through the cell, which make up the cytoskeleton, making them turn different colors so that we could actually see them. What does the cytoskeleton do? Well, it provides internal support, helping the cell to keep its shape. It allows organelles within the cell to move, such as the organelle shown over here on the left. This is probably a vesicle of some sort that's en route, maybe going from the ER to the Golgi, and it will walk along this long path, and the actual path itself is one of these strands inside the cytoskeleton. It can also help the whole cell to move and divide. Like in this sperm cell, the flagellum, the long tail that comes off of it that beats back and forth, moves thanks to the cytoskeleton. These tinier, small tails, called cilia, coming off of this paramecium, also move thanks to the cytoskeleton. And when a cell is dividing, it separates its chromosomes and other organelles, pulling them to either side of the cell, also using cytoskeleton. Last but not least, we have the energy processing organelles, your good friends, the mitochondria, which release energy from sugars like glucose and put it into ATP so that the cell can use it. That's by a process you already know called cell respiration. And then the chloroplast, which runs photosynthesis. That magical process that harvests sunlight energy to make sugar out of air and water. In conclusion, if you take the right combination of any of these organelles and you put them together inside some cells, and then you take those cells and put them together, you could make any of the diverse organisms on this planet, whether it be a plant, or a fungus, or a bacterium, or an archaeobacterium, or a protist, or even an animal.